Hi, my name is Cody Cardenas, and I'm a graduate student at The Ohio State University, Department of Evolution, Ecology, and Organismal Biology. I'm here to present my research on the population genetics of a new species of Panamanian fungus-growing ant. For my presentation, I'll give you a brief introduction to the fungus-growing ants, and then I'll introduce the research that I've done with the new species. Fungus-growing ants have a fascinating symbiosis with the fungus garden. They bring organic material back to the nest, process it, and integrate it into the fungus garden. The ants then get to eat the fungus garden. The fungus gains dispersal and protection from the ants. This relationship has allowed the fungus-growing ants to radiate throughout the New World. There are over 240 described species, with many left to be discovered. The species that I study belongs to the new genus Mycetomoellerius, which came out of the genus revision of Trachymyrmex just last year. In fact, I'm describing a new species, Mycetomoellerius cfziteki, or little black. I'm using an integrative taxonomic approach which uses morphology, behavior, and chemistry. But this new species isn't so new. I found that over 60 articles have likely studied the new species, and not Mycetomoellerius ziteki, or big red. Little black is a species I'll be using for my population genetic research. Population genetics allows us to take discrete units, such as species, and understand how they are all interacting at the individual level, or population level. Population genetics also allows us to consider the effects of selective pressures. We can think about how populations might shape or be shaped by their fungus gardens. There are also a lot of parasites in fungus-growing ant symbioses, fungus that attack the garden, other ants that live obligately in the nest, and parasitoid wasps that attack the ant larvae. But we would be remiss if we didn't consider the environment of Panama where we find these ants. We can consider the comparably different forest types on the Caribbean coast versus the Pacific coast throughout Panama. There is also the deforestation and flooding that was necessary to construct the Panama Canal. Circled is the 42,000 hectare lake Gatun in Barrow, Colorado Island as a star, both of which formed as a result of the flooding. This photograph gives a sense of the impact this had on the forests. There was also 100 years of pesticide use and swamp drainage to help keep mosquitoes at bay. Since the 1980s, the Panamanian government has also been protecting forests. These shaded areas represent natural parks. You can also see that there are a lot of waterways, creeks, and rivers in Panama. It's these creeks and rivers where we expect to find a little black, as they tend to nest on vertical embankments. Here you can see a few incipient nests and an example of a creek in the Panama Canal. These creeks represent an expansive network of ideal habitat for the ants. Alternatively, there aren't nearly as many vertical embankments in the forests. Thus, creeks may act as corridors. Reproductives are also predicted to disperse only short distances, so we expect a sort of creek fidelity where nests are established on their natal creeks. Here you can see the network of creeks that we are calling watersheds. For example, Birdplot, Rio La Seda, and Rio Friolito all share the same watershed. So, for my project, I have 3 research questions. Are there distinct genetic populations in the Panama Canal zone? Does isolation by distance act on these populations? And is geography influencing these populations? Relating back to our idea of creek fidelity. I am going to focus on a few key results, so if you have any questions, please feel free to reach out to me on social media. We used a double digest restriction site associated DNA sequencing approach, followed by a bioinformatic pipeline that allowed us to identify 1,100 single nucleotide point changes, or SNPs, from 74 colonies. One of the filtering steps we used was to remove loci that don't meet Hardy-Weinberg equilibrium expectations. We see that for these filtered samples, our Nays and Hendrix fixation indices both agree with approximately 0.11, and our inbreeding coefficient is below 0. This suggests that there is a single population with some genetic differentiation, but not enough for distinct genetic populations. We also performed a pairwise comparison of fixation indices between creeks. Fixation values are on top and kilometers are on bottom in bold. Comparing Birdplot south to Parque Natural Metropolitano, or Metro Park, you can see that the fixation indice does increase. This change in our pairwise comparisons could be from isolation by distance, and this is supported by our mantle test. We visualize geographic distances on the x-axis and genetic distances, or the fixation values, on the y-axis. As we increase in geographic distance, we do tend to see an increase in our estimated genetic differences with FST. We also used clustering methods to estimate the number of populations in the past from our samples. Because we have isolation by distance in our data, we use the clustering method construct, which models populations with an explicit assumption of isolation by distance. We see that for population models of 2 through 4, based on the allele frequencies of our creeks, there appears to be one population. We also used ANIMOVA, an analysis of molecular variance, to see if there is an influence of geography on our data. Overall, we see that comparing all colonies explains the most genetic variation, what we expect in a panmictic population. But we do see small amounts of genetic variation at the creek and watershed level. The phi statistic is also comparable to the previously shown fixation indices, and these also support some genetic differentiation, but again, not enough to indicate distinct genetic population. Lastly, we use the program Colony, which shows contemporary sibling relationships. We have colonies listed on the both x and y axis and squares representing creeks. If a colony has a sibling on a creek, it would fall in that square. And we see that in general, most creeks have lots of siblings. But there are some long-distance relationships, particularly between Metro Park and Pipeline Road, approximately 25 kilometers apart. Now, let's break down what all of this means. Using neutral loci, we found a single panmictic population. This result shows there is gene flow throughout our entire sampling scheme in the Panama Canal zone. While we did find an effect of isolation by distance, this is a realistic expectation and a normal feature that is often found in populations. We did find some effect of geography in our data, but it was not enough to differentiate our populations. Some variation in our data could come from selective pressures. For example, it has been documented that some creeks can be more heavily parasitized than others. Alternatively, this variation may come from creek fidelity. And we did find that these colonies generally have siblings on the same creeks. Some neighboring creeks that are close by also have a higher likelihood of having siblings, but we did see some occasional long-distance dispersals. However, when we were collecting the ants in the Panama Canal zone, we only ever found them in moist forests. Also, we only found colonies in more disturbed habitats, like the southern parts of Pipeline Road and Soberania National Park. According to Condit et al. In 2011, compared to areas like BCI and northern parts of Pipeline Road, these are more disturbed and secondary sites. This may suggest that Little Black could have preferences for disturbed sites. However, more work needs to be done to understand the biology of the species, such as understanding the flight ecology and determining differential dispersal between male and female reproductive castes. We can also compare Little Black to other fungus-growing ants and non-fungus-growing creek-dwelling species found in Central America to better understand how creeks shape the populations of these species. This research was made possible by my department's research experience for undergraduates, which allowed Andrew to generate preliminary data. The Smithsonian Tropical Research Institute for allowing us to collect samples in the Panama Canal zone. My advisor, Dr. Rochelle Adams, Behavioral Ecology and Evolution course in Panama, and a lot of great funding resources. I also would like to thank everybody that's supported me in my development as a young researcher, and everybody in the Adams Megalomyrmex Ant Lab at OSU. Thanks for listening to my presentation. If you have any questions or comments about research methods, my bioinformatic pipeline, things you didn't see, please reach out to me on Twitter or Instagram.